The human microbiome, a complex and diverse organ system, is made up of bacteria, archaeoviruses, which are typically bacteriophage, and fungi, along with their genetic material that reside in and on the body. Each region of the GI tract has its own unique microbiome that differs in membership and function. Many factors contribute to the unique composition, including route of birth, age, infectious exposures, lifestyle, diet, medications, ethnicity, geographic location, and genetics. Assembly of the microbiome starts immediately around birth and develops during the first 3 years of postnatal life. This is when microbial signaling influences the education and maturation of the immune system and internal organs takes place. The microbiome evolves throughout life and contributes to both health and disease. The microbiome and its complex interactions with other organ systems play a key role in protective, metabolic, immunologic, and physiological functions that are crucial to human development and health. Microbes produce vitamins, synthesize amino acids, and carry out the conversion of primary to secondary bile acids important for the anti-inflammatory signaling. Oligosaccharide fermenting bacteria metabolize dietary fibers into short-chain fatty acids that provide energy to colonic epithelium and play key roles in immune regulation and metabolic homeostasis. Pathogens must compete with the environmentally well-adapted microbiota for nutrients and attachment sites in the gut lining, making it difficult for these pathogens to colonize, a process known as colonization resistance. The microbiome also demonstrates therapeutic benefits such as the treatment of recurring C. difficile infection by intestinal microbiota transplantation to reintroduce or boost helpful organisms. Similarly, studies have also looked into the potential therapeutic benefits of probiotics, prebiotics, symbiotics, and postbiotics for many GI and liver disorders, such as IBD and chronic liver disease complications. In the U.S., most probiotics currently on the market are either foods additives, or dietary supplements. Current regulatory policies allow manufacturers of probiotic foods and supplements to make structure, function, or health-related claims that are not related to diagnosing, treating, curing, or preventing any disease. Further research is needed to substantiate the benefit of probiotics for individuals managing a health condition. According to the AGA's recent clinical practice guideline on the role of probiotics in the management of gastrointestinal disorders, there is evidence to support the use of probiotics for some GI disorders, but insufficient evidence for others. In general, more clinical data is needed. Diet impacts the gut microbiome in both health and disease. Infant diet is pivotal in the compositional development and diversity with variation between breastfed and formula-fed children. The resources from our diet maintain our health and homeostasis. However, animal-based, high-calorie, high-fat, and processed sugar diets, low in dietary fiber, have led to decreased microbial diversity and impaired function. Loss of dietary diversity in the elderly has had similar effects on the microbiome and is associated with increased risk of inflammation and frailty. Thus, diet can be one of the simplest microbiome-based interventions. While a healthy microbiome works symbiotically to facilitate health, some microbiome patterns are associated with inflammatory bowel diseases, irritable bowel syndrome, liver disease, obesity, and type 2 diabetes, to name a few. It is unclear if such dysbiosis is a cause or consequence of the disease. Gut dysbiosis in liver disease is associated with accelerated fibrosis and cirrhosis development through an upregulation of inflammation and contributes to further complications such as hepatic encephalopathy. Patients with IBD exhibit reduced microbial diversity, decreased relative abundance of firmicutes, and an increase in protobacteria. Loss of microbial diversity is associated with loss of functions essential for maintaining and regulating the immune system. The microbiome is also associated with frailty and disease in the elderly. Dysbiosis causes a decrease in mucin and starch degradation, essential amino acid synthesis, and nitrogenous base and vitamin synthesis. Shifts in the microbiome among older people can arise from cultural factors and environments related to changes in diet and activity with aging, living, and care settings, and exposures to medications. Gut dysbiosis caused by these shifts have shown to stimulate pro-inflammatory mediators and induce systemic inflammation. This may be associated with neuroinflammation, cognitive impairment, and other neurological disorders like Alzheimer's disease. The microbiome composition may also have both preventative and causative effects on cancer of the GI tract and other organs. Evidence shows implications for carcinogenesis, response and toxicity to therapy, anti-tumor immunity characteristics, and clinical response to immunotherapy. Understanding the association between disease and the microbiome can inform new methods for risk stratification, prevention, and treatment. New research aims to increase lifespan as well as improve and maintain quality of life through microbiome modification. This includes targeted microbial metabolite strategies to improve metabolic disorders like obesity and type 2 diabetes. One of the most complex unanswered questions is how to define a healthy microbiome. Many factors contribute to this. For example, there is no single microbiome composition required for health. Research is limited by the technology currently available, and more widely representative and longitudinal studies are needed. There is also little known of the contributions to health and disease by viruses, fungi, phage, and archaea. Advances in our knowledge will pave the way for future clinical applications aimed to prevent and treat disease. For more information on the microbiome, please visit gastrojournal.org.